All right. So everyone here took the test over the weekend, and I can tell you that the average was high enough that it made me uncomfortable. The average was like 90%. And it's like, oh, is that because I've gotten so much better at teaching this material? Or is it that, anyway, I got to tell you, it made me a little bit nervous that maybe people were talking to friends that had seen the test earlier, and I don't know. But so I'll just say, you are supposed to work in the test entirely on your own, right? Not to share information, et cetera. So I will choose to think, and I even thought about, well, maybe we should go back to having, everyone take it at the same time. But then that gives me fewer classes to yak at you. And you know how much I love that. And also, I said in the beginning we'd do it this way. So, okay. So, they don't dock my pay or anything if everyone gets A's, right? So it's not that. So, questions about the exam. Another one coming up, like 7 or 8 lectures from now. Yes. The final will be a cumulative final.
That was on this class to on and the
Yeah, so beyond any doubt, there'll be some questions from test 1 on the final, and for each of the tests. Yeah. Other questions, excellent questions like that. Yeah, Abdur.
For the quizzes so that we see the exam.
Oh, let's say it again.
Feedback open for the quizzes so that we see what.
Well, feedback. I don't know. I don't. We don't know. I don't normally do that. But if you make an appointment to and Antonio's not here today. He's not feeling well. But if you make an appointment with Antonio to go over it, yeah, that would be fine. And of course, many of the questions come from Kahoot quizzes, and those answers are all posted. Good. You okay? All right, we're trying our best to build the most complicated machine in the universe. And in chapter 1, we figured out how to start making an organism where different cells communicate with each other to direct each other in cellular differentiation. And then in chapter 2, we figured out how to take a starting point, and from that starting point, have a wave of induction like that be such that you end up with a body that's properly organized, right? With the head where it needs to be, the tail where it needs to be, legs, et cetera, et cetera. Then in chapter 3, we talked about how you have to populate this brain. So we talked about neurogenesis, and it turns out shortly, at almost as soon as a new neuron is born, it starts moving around. And so now we're going to reach the point where eventually we have a nervous system that has about the right number of cells in the right places. And now the process has to begin of differentiation within each of those areas so that you get the right combination of various cells. And it turns out that, again, it's cell-cell communication all the way. And remember from the clones that we studied in the spinal cord, remember, even at the very last division, 2 different cells may take on a different fate, which emphasizes, again, we're not doing it in the mosaic specification of cell fate the way worms do. Rather, the cells remain plastic right up to that last division, at least some of the time. And so how do they get their different fates? How is it that you don't have only motor neurons in the spinal cord or only glia? Well, it turns out that just as with the delta notch system that we saw in flies, it turns out that those sorts of interactions are going on so that the cells are constantly, if you think of it as competitive, they're constantly competing with one another to see who's going to take which fate. You can describe the same thing by saying the cells are constantly cooperating with each other. Oh, you're going to be a motor neuron, I'll be an oligodendrocyte instead, right? But either way you conceive of it, what it requires is that the cells have to be talking to each other, and they have to be listening to each other. okay? So we're going to talk about some model systems where we know what's going on. And I should say that we're emphasizing model systems because that's a big problem, right? 86 billion neurons and maybe 10 times that many glia in your head, you know, figuring out how each one of them got a very specific designation, a very specific shape and function, that's we're never going to get there, right? But in these model systems, you can see how these things might work, and that in principle, at least, you could see how you could end up with a nervous system with 86 billion neurons and no two of them are alike, right? Really hard work. And as often as the place, bless you, as is often the place where we made a lot of progress was in fruit flies, again, because of the power of getting to manipulate a genome, having a genome so well mapped out, and having a relatively simple organism. And a big breakthrough came in understanding the differentiation of the eye in fruit flies, the ommatidia. So as you know, like most insects, flies have a compound eye. And if we were to look at these compound eyes, with each one of them consists of a more or less hexagonal column. And they're all packed together. And hexagons, it turns out, is the most efficient way to get the most highest density packed together, as honeybees have told you several times. And in each of these ommatidia, and if we could talk about one being an ommatidium, in each of these ommatidia, there are going to be 7 different photoreceptor cells, each slightly different from one another in terms of what they do and how they do it. Now, each ommatidium also has other cells going to make the lens, and other cells going to be support cells, and other cells that are just going to be backdrop to keep light from passing through, stuff like that. But we're neuroscientists, so we care the most about the neurons. And when people started studying these, I want to point this out, that they came up with a terminology where they would refer to each receptor cell as either R1, R2, R3, etc., for R8. And what's fascinating about studying Drosophila is sometimes you can watch embryogenesis going on right there under the microscope. And so there comes a point when there's an optic disc formed. And at first, the optic disc is, formless, is a void without form. And then what happens is, starting from posterior to anterior, a wave happens where you can see a physical ditch appear in the eye and sweep its way anteriorly. Everyone got that? And in front of the ditch, the cells are totally amorphous. And behind it, they're already organized in a recognizable hexagonal shape. So this furrow is moving across what's going to be the eye and leaving structure in its wake behind it. So it's called a morphogenetic furrow because it's generating right? It's generating structure, morphology behind it. And that morphogenetic furrow is being triggered by posterior cells that are secreting hedgehog, not sonic hedgehog, but a relative hedgehog. And the posterior bit, let's get the picture here. The posterior bit at here, they induce the cells there to start making hedgehog themselves. And it's being secreted. Hedgehog is a secreted protein. And so what's happening is there's a set of dominoes. You start secreting hedgehog, and so you start secreting it and telling your neighbor, who in response to that starts secreting it, and so on and so on. And pretty soon the whole disc, the cells are all expressing hedgehog. Right? And back here. And so, yeah, what else do I say? Yeah, and the sonic hedgehog the hedgehog in turn kicks off expression of another protein called DPP. And the DPP is going to have some cells start expressing proneural genes, the same old proneural genes that we talked about with Notch and Delta. And as in that case, what's going to happen is there's one cell in the middle of that induction that probably gets the largest initial instruction, and it's the one that starts expressing the proneural genes first. And instead of it suppressing proneural genes and all of its neighbors, once it gets started, it starts inducing its neighboring cells until there are a total of, I've been saying that, a total of 8 photoreceptors. Oh, and yeah. How's that? Good.
Learn that galaxy.
Pardon?
What she saw the first time.
I don't know, but something has done that because it's being expressed at those cells that are at the posterior edge of the optic disc. Probably an earlier induction, right? A la C chapter 2. Yeah. Justin?
VTT is a BNC
Homolog. Yeah.
How come it turns itself pro neurons?
It's a great question. So here's the same signal that in other cases suppresses proneural expression. Now it's going to promote proneural expression. And what's the difference? Well, these cells have a different developmental history than in the early stages, right? And they've been places and done things, right? Including express lots of transcription factors and in a particular sequence. And so they're primed to respond to it differently. And that too is dead common. So we go back here to look at this, we'll see. And I was thinking today, I wish I'd reversed this in the second edition. I didn't. Anyways, because it's kind of confusing. Okay, so the furrow starts here and moves across, and here you can see the morphogenetic furrow. And maybe you can see that in front of it, ignore the green marker, it's just a green marker. In front of it, everything's kind of mushy, and behind it, can you make out the hexagonal pattern already, right? So formless, a void without form, formless without, what's the phrase? Oh, well. And behind it, structure, morphology. And if we were to look at the ommatidia here in a line, we'd see those that saw the morphogenic furrow first, they are all, those that haven't seen the morphogenic furrow, they're all there, all 8 cells, but none of them are expressing proneural genes yet. And the ones that just had the morphogenetic furrow pass over them, and those one cell wins some sort of competition, beyond doubt, and it's the cell that starts expressing proneural genes, and it's going to be receptor 8. I'm sorry it wasn't receptor 1, but they numbered the receptors before anyone knew anything about the development. And what's going to happen is cell 8, after getting ahead of its neighbors, is now going to start inducing 2 neighbors to make the receptors that are eventually going to be 2 and 5. And then, further yet, 2 and 5 will induce the development of 3 and 4, and then 2 and 5 will then induce the development of 1 and 6. And there comes a stage where every receptor cell is on its way to becoming a neuron except one, the cell that's fated to become receptor number 7. And eventually, it too starts expressing proneural genes and goes down the pathway becoming a photoreceptor. How's that? So far, so good. And in a pretty heroic effort look at that, in the, I guess the 1980s, with a screen of every mutation they could find and a screen where they would look at the head of each of 100,000 some odd different flies. they isolated a mutation where receptor 7 was missing. And so they named that gene where a loss of function gets rid of receptor 7. They named it Sevenless. Everyone see the logic of that? And they reasoned that it must be that Sevenless, that the defect is it's not listening to the signal, right? So they cloned the gene, sequenced it, which used to take a lot of time in those days, and eventually realized that it was a transmembrane, it had a transmembrane domain, which means it's embedded in the membrane, and that on the interior surface, the surface that's going to be inside the cell, in the cytoplasm, there was a tyrosine kinase. A kinase, right, an enzyme that phosphorylates other proteins. And in this case, it's one that only phosphorylates tyrosines, and not every tyrosine, just tyrosines in particular positions in a protein. And biochemists have been studying these tyrosine kinases for quite a while, okay? And so they realized that it was an RTK, that is a receptor tyrosine kinase. I'm sorry that that's the order that it's there. And once they started looking for other such things, they found that it's an enormous superfamily. So you have loads of RTK genes in your genome, otherwise you wouldn't be here. And they play a lot of different roles, but it turns out that, remember we talked about ephrins and ephrin receptors? Well, the ephrin receptor is also a tyrosine kinase, a member of the RTK superfamily. So Sevenless, which one is Sevenless? Where is it? Maybe it's this one. Sevenless, once it has the defect, it can't respond to the extracellular signal and doesn't then do the tyrosine kinase thing. It doesn't phosphorylate the right proteins inside, and it never goes any further down the pathway and never becomes a neuron. See that so far? And we're going to run into many RTKs in the future because remember, the whole thing about development is cells are constantly talking to each other, and this is a major pathway by which cells talk to each other. It is a receptor protein, so this is how one cell can affect its immediate neighbor, one that it has its membranes up against. So among them that we'll see, we've talked about fibroblast growth factor. It turns out the FGF receptor is an RTK. Later, we'll talk about TRAC receptors that respond to neurotrophic hormone proteins, that is, proteins that keep cells alive. They're a member of this family. We'll run into MuSK when we talk about development of the neuromuscular junction. I've mentioned ephrins already, etc. And I just want you to know that it's a large superfamily and that all the RTKs have a tyrosine kinase in the intracellular side. I love this figure because it shows what similarities they have and differences, but I don't want you to remember any of that detail, right? Do not tell me how many, cysteine-rich aspects are in which RTK receptor. Nobody remembers that stuff except people that cloned and sequenced that particular gene, right? I don't want you to do that. You okay? So don't be afraid of this figure. It's just telling you what RTKs are. And a breakthrough happened when they found the mate of RTK. Uh-oh, have I done it? So they found another mutation that also caused 7 not to appear. And this gene, when they isolated it, was only expressed by receptor 8. None of the other cells expressed it. And so they reasoned, receptor 8 secretes a protein, and when it hits receptor 7, as long as its Sevenless gene is working properly, that cell will then become receptor 7. I'm sorry it's confusing that the gene's called Sevenless, because even though when it's working properly, it ensures there's going to be receptor 7, right? They named it after it, what happens when you knock it out. And so they call this other gene Bride of Sevenless as boss. Again, a little joke from Larry Zapersky at UCLA. I thought it was funny. And so receptor 8, there comes a point when it expresses boss, and it has some effect on the other cells, but it's crucial for R7 to develop. So here's an example of how in a complex of cells where they have to take on different fates, they are talking to each other and arranging to make sure every ommatidium has exactly one of each of the 8 different receptors. And by the way, when they found the sevenless mutation, it was a very specific defect in the behavior of the animals. It turns out receptor 7 is important for seeing UV light, which flies do and you don't. And they could show that now they couldn't detect those wavelengths. How's that? So more of that delta notch stuff, and then more specific stuff, right? Okay. In going back to vertebrates, that delta notch thing, we know that's going on in the vertebrate nervous system too, and I've alluded to this before. But what's going to happen is cells have various amount of delta that they're expressing. They're all expressing notch, the receptor to delta. And those that tend to have higher expression of delta to begin with start inhibiting their neighbors so that one cell becomes the neuron and the others become glia. And again, that's happening because they have their membranes touching one another. And when it's all over, you have some cells that are going to become neurons and others are going to become glia, and there are different kinds of glia. And, there are different little arenas throughout the neural tube where this discussion, competition, cooperation is going on. And enough of them are there that when it turns out it all works out. I want to point out that this is a stochastic process, by which I mean that which particular cell is going to become a neuron and which is a glia, looking ahead of time, you can't really predict which one is going to be that. So you can't predict on the level of an individual what will happen. And yet the rules must be such that when you step back, it'll turn out that the number of neurons and the number of oligodendrocytes and the number of astrocytes is pretty much the same from one individual to another. So this competition, cooperation, discussion that's going on will end up with about the right number of cells. And so that's what I mean by stochastic. The parameters must be there to get the right proportions when it's all done, but you can't predict ahead of time who's going to take on what fate. Why? Because they're hashing it out among themselves.
We just count if one has what's delta.
You would think that in theory, at least, that it could be possible, right? That you could see that this one has a bit more. But you can also think about situations where what if they have exactly the same amount? And then it might just be a matter of who works fast or whose notch happened to run into its neighbor's delta, right? So it's up for grabs, like elections, right? I mean, things are changing all the time. I mean, and I will say, in that sense, this model of development is very small d democratic, right? They're all yakking at each other. Everybody's listening. Everybody's talking. Everybody's responding. And it all works out. If we go back to the nervous system, then you can see that there's this decision tree that's happening that's going to specify everyone's fate. So early on, remember, it was the BMPs, bone morphogenetic proteins from the epithelium, that were inducing more epithelium. And it was the organizers like noggin and cordon and follistatin that were getting some to start taking on the neural fate. And so that's how the neural tube gets formed. And then once the neural tube gets formed, we have these inductive features from up dorsal and ventral, so that every cell is getting a slightly different combination of these things. And therefore, the competition that's going on there is a little different. So it's only going to be in the floor plate that, because of the influence of sonic hedgehog from the notochord, it's only going to be there that any cells start competing to be who's going to be a motor neuron, not in the dorsal horn. And so that's why all the motor neurons are going to end up in the ventral horn, which is handy because their axons have to go out the ventral root for all this to work. I'm not showing it here, but also remember that this is a dorsal ventral continuum, but there's also a rostral caudal continuum of those various factors we talked about last time, such as retinoic acid, etc., FGFs, Quint, etc. And so every portion of this neural tube, cells are getting a slightly different signal about what genes they should be expressing, and that will end up causing them to take on slightly different fates. Now we've added another bit of complexity, right? So that even within the neural portion, some are using delta notch so that they become neurons, and others become glia. And of the glia, some are going to become oligos, and some are going to be astrocytes. And so people have identified the things that are going to start pushing them to become oligos or astrocytes. And we'll talk some more about neuroregulin. We won't talk much more about 1 and 2, but we'll talk about neuroregulin later today too. So let's go back to talking about motor neurons. I show this here just to show that motor neurons aren't all alike. Different motor neurons innervate different muscles and therefore have different functions to do, and of course are going to get different synapses once the brain starts sending its axons down the spinal cord. And here is just a nice pretty picture where each of the color represents a different Hox gene. Remember the Hox genes in mammals, Hox on 4 different chromosomes, Hox A, Hox B, etc., that are being expressed in different portions of the ventral horn. And this is the Hox gene that's also expressed outside the ventral horn. Eventually, there will be 4 different kinds of motor neurons in this bit of the spinal cord. And again, I don't want you to memorize this, but if you keep track of them during development, you'll see that early on, they're all expressing some genes that are called islet 1, which then causes the expression of islet 2. Who cares why it's called that? And what happens then is there's a choice point, and some keep expressing those 2 and also express another one called limb 3. Others only express those 2. Others express limb 1 and IL-2. Others only IL-1, right? And so each of these pathways is directing the cell to differentiate in such a way that will play a different kind of motor neuron. There are the motor neurons in the medial column of the spinal cord that in general tend to innervate the muscles of your axium, your trunk, rib muscles, abdominal muscles, stuff like that. So that's your axial musculature. It's along the axis of your body. And those motor neurons tend to be in the medial half of the ventral horn. And it turns out that those cells are being induced to go down this pathway from FGFs that are being released by the muscles. So again, cells are talking to each other, and they're taking on a different fate depending on who they've been talking to. And the important thing is that sometimes it's the target that tells the cell what to do. Then in the lateral motor column, that's where all the motor neurons to the limbs live. And even there, those that are in the medial subdivision tend to be playing a role in medial, in the abductor muscles, and those in the lateral tend to be important for the dorsal limbs. And they've gone through a different set of expression and therefore, take on different fates. And then other cells are going to be sort of high in the ventral horn. That's where the sympathetic preganglionic cells live. They're going to be sending out and innervating cells in the ganglionic chains on either side. Again, I don't need you to remember the details of what signals they each have, but I do want you to know the 4 different kinds of motor neurons. And I want you to know that they have a different developmental history in terms of the transcription factors they've been expressing. And what have we got here? Oh, yeah, just to say, because we'll run into them again, the limb and islet genes, they happen to have parts in their domains that are known as zinc fingers. It's places where the protein folds to make these fingers, and it can't do that without a zinc ion around. Okay? And what people learned pretty early on, actually, in study of steroid hormone signaling, is zinc fingers are a way for the protein to latch on to the DNA and start affecting gene expression. So when you see genes with zinc fingers, you automatically know they're a transcription factor. And I say that because we'll run into zinc fingers again. Yeah, good.
I don't get what parts of the body, the ventral, left, and dorsal.
Well, let's see. It's not it's the dorsal half of the limb. So there's the dorsal half of the arm and the ventral half of the arm, and the same with the legs.
So like bicep and then forearm, or like
no. Like bicep and tricep, right?
Okay.
Biceps on the ventral surface, tricep is on the dorsal surface, and triceps tend to go away, and biceps tend to bring the limb closer to the body. Okay. I wish I hadn't even talked about abductor muscles. Forget that. Dorsal ventral, that's all you need. But it does mean the whole limb, not just, because that's the dimension we're talking about, dorsal ventral, not, proximal distal. Okay. And these Hox genes are going to be important too in the rostrocaudal distribution of different types of motor neurons, where here's a map that's showing you various Hox genes, which ones are being expressed in different parts of the rostrocaudal spinal cord. And remember, how do they know whether they're rostrocaudal? Because they've got those rostrocaudal cues that are also making a continuum. And again, I don't want you to remember, that's HOX6 for the lateral motor column and the cervical region, et cetera. I just want you to know that they're different, that they have a different pattern of Hox gene expression. And these are, Hox genes, not just homeobox genes, but Hox genes, right? Which in fruit flies cause those bizarre structure swaps. But in vertebrates, because there's more redundancy, tend to have more subtle effects when they're mutated. But the fact that in mice, that's where all this got worked out, the fact that they're being expressed in different places in different motor neurons is a clue that they are going to direct gene expression in that cell to take on a different function. And here, I'll just point out, sympathetic preganglionics, they tend to happen in the thoracic spinal cord, right? Sympathetic nervous system used to be called the thoracolumbar nervous system because they get a little bit in the lumbar system, but the sympathetic preganglionics are going to be here, and in the cervical region, you're going to have lateral motor column because there's a limb there. In the thoracic region, you don't have a lateral motor column because there's no limb, right? It's only axial motorons. And again, in the lumbar, again, you will also have, I mean, we're not showing it here, but there'll be a lateral motor column in the lumbar region too. Clearly. Time for Kahoot.
I'd agree.
Okay. So now we're going to go back to neural crest cells and take up the question of differentiation of neural crest cells as they start migrating. And one of the things that made our understanding of neural crest cells easier was that in chick embryos, right? Remember where you can just take a pair of scissors and open up the top of the egg and see the embryo and then manipulate it, sort of thing you can't do in mammals, where people found that you could take a chunk of the neural tube and move it from one place to another. And to keep track of which was which, they would take, say, a chunk of neural tube from quail and put it into a chick embryo. And there's just a quirk of quail cells is that they have really dark nuclei. So you can always tell which is a quail cell and which is a chick cell by that method. I didn't include a picture of this. Well, I guess there is in the book, but it isn't important, right? You can just tell them apart. And what they would see is they could end up with a chick with a quail spinal cord segment within a chicken body otherwise. And when they do these transplants, inevitably, at the stage they're doing it, they also move neural crest cells as well. So another way that they knew this worked was that they could end up with a chicken like this, where there's a band of brown cells, because remember the neural crest cells make melanocytes. And quail neural crest cells make melanocytes, and they happen to use white leghorn chickens because they're common and cheap and hardy, whereas the rest of the body is white. And what that tells you is that the neural crest cells are going to make melanocytes. They mostly just go straight out. They don't seem to be moving up and down very much, right? So they know where the midline is, and they just head out and will eventually populate the skin. Oh, and here's a picture. So in these cells will have a really dark nucleolus. Those are quail cells, and those nearby that don't have a dark nucleolus, they're chicken cells. So you can see at the microscopic level who is who. And to go back here, this meant that you could ask, once the neural crest cells form, have they already been specified as to what sort of fate they'll take on? Because it turns out that cells in this part of the neural tube, they're going to make the sympathetic ganglia that use norepinephrine as a neurotransmitter. Whereas the neural crest cells down here, further along at the sacral region, they're going to make neural crest cells that become the autonomic ganglia and therefore use acetylcholine for a receptor. As a transmitter. So, in terms of terminology, this is useful. These sorts of things where you take different species and mix them together, they're known as a chimera. And you can sort of see obviously why, right? It's a chimera, as in Greek monsters that were several different animals put together, a dragon and a lion with a dragon's tail on a lion's body and an eagle's head and stuff like that. Sometimes, though, you can have chimeras that are the same species, and that's not unusual in mice. And so where you take some cells from one mouse embryo that has one genotype and mix them together with cells from another mouse embryo with a different genotype. And you can end up with an animal where some parts of their body are one genotype and other parts are another. And those are called chimeras too. The easiest way to keep track of what's a chimera versus a mosaic and other things will come up is chimeras tend to have 4 grandparents. Right? Each individual has 4 different grandparents. How's that? So what happens when they did these sort of transplants, when they took cells that if they'd left them alone were going to become noradrenergic and moved them and put them in a lower part of the chick, did they stubbornly insist on staying adrenergic? The answer is no. Put in this new location, once they started migrating out, they took on the fate of, I better get this right, they took on the fate of the adrenergic fate that's appropriate. And likewise, if they took sacral portions from the quail and put it in the thoracic region of a chick, I had this wrong. Oh, sorry. So the distinction is this is up by the head, right? And this is the thoracolumbar region that's going to become adrenergic. And wherever they put them, they took on the fate that was appropriate for their new location. So at least at the time when they took out the chunk and moved it, the neural crest cells didn't know what their fate was going to be yet. And so they must have gotten their instructions once they started migrating out into the periphery. So again, they're plastic. They can take on the fate that's appropriate for their new position because they're going to get different instructions once they start migrating out and therefore take on a different fate. How's that? So you all learned in 301 or 302, right, that sympathetic fibers release norepinephrine and parasympathetic fibers release acetylcholine, hopefully, right? That got discussed at some point. Were you also taught that there are exceptions to that? There are some sympathetic cells that release acetylcholine in the postganglionic cells, specifically for sweat gland. So, oh, nope, I'm not ready for that yet. Sorry. And I'm sorry Antonio's not here. You'll have to listen to the recording. It turns out that this increased plasticity of neural crest cells is sometimes a bad thing. So it turns out Hansen's disease, once known as leprosy, it's caused by a bacterial infection. So today it can be treated with antibiotics and people can be cured of it. But before that day, what's interesting about the bacterium is they tended to infect Schwann cells. And remember, Schwann cells are derived from neural crest cells. And they infect the cells at one point and then just sit there and do nothing but multiply without ever leaving that particular cell. That's the typical natural history of them. But eventually, if there's enough leakage, the immune system notices that something's going on here and starts attacking that region, and there's inflammation that happens. And as the nerve gets inflamed, some of those axons die. Well, it turns out the signal for Schwann cells to form is a signal that's embedded in axons called neuregulin. Remember, we ran into neuregulin. We're talking about earlier stage of development and differentiation. So neuregulin is a membrane-bound protein that neurons put in their axons. And the neuregulin, when it's recognized by neural crest cells, turns them into Schwann cells. So the neural crest cell is moving along, it bumps into an axon, and the axon, because it's coated with neuregulin, starts pushing that neural crest cell to become a Schwann cell, and the same signal will also cause it to myelinate the axon. And it turns out axons decide how thick their myelin sheath is, because the more neuregulin they express, the thicker the myelin sheath will be. Another example of cells communicating with each other to make sure, for example, that all your alpha motor neurons, where you want really fast responses, that they're all great big axons with loads of myelin, so conduction is especially fast. Like those cells that signal to you that you just put your hand on a hot object. As a reminder, that stimulates nociceptors that have big A delta fibers, right? And so you get the signal right away, and the reflex happens, and you withdraw your hand. And then after your hand's already withdrawn, then the other set of pain fibers, the nociceptors that use the tiny little C fibers that are not so myelinated, that's when you start getting their signal, and it aches for a long time. So the first signal is get your hand away from there. You need big fat fibers. And the second signal is don't do that again, in which case slow-acting fibers will get the message across. Right? Anyway, the first pain fibers, the reason they have so much myelination, the A delta, is because they've given more of a neuregulin signal to the Schwann cells than the C fibers have. Okay. Well, so what happens is inflammation happens, the axon's lost, and so that neuregulin signal to the Schwann cells is lost, and some of the Schwann cells de-differentiate. They remain plastic even this long, and now they start migrating again. And they take on other cell fates, including melanocytes and adding to muscle fibers. Unfortunately, they're also carrying the bacteria, and so now there's an infection that's being detected by the immune system in these melanocytes, and it starts attacking it, and eventually you have loss of sensation there, and eventually loss of circulation, and fingers start falling off, and toes start falling off, and tips of noses start falling off, all that stuff. Yeah.
So is your immune system only able to detect them once they become melanocyte?
Pardon?
Your immune system can only.
What's interesting, apparently while they're migrating, it doesn't seem to care. But once they start differentiating into melanocytes, then something about them being infected means the immune system notices them right away. Whereas when it was latent in the Schwann cells, I mean, they tend to be there for years. Yeah.
Is it atypical for cells to be plastic for that long? And is there a reason that Schwann cells are?
It's a great question. Just as you can only measure commitment in terms of the challenges you give a cell, you can only measure plasticity in terms of what you did to make them decide whether they could change their minds or not. But I will say that in general, it seems like neural crest cells remain plastic a long time, pretty much forever. Yeah. And that's, that I don't know, they're special. To finish the story of Hansen's disease, and we'll run into this again too. The neuregulin is binding to ERB, another RTK that's on the neural crest cells, and we'll run into ERB again later. This neuregulin from the neuron, ERB from a target. That's going to be important when we have the neuromuscular junctions develop soon.
Neural cells also made from C acid.
True.
But why did they show that to be muscle?
Yeah, they're actually providing support cells within the muscle. So good call. I don't think they've developed into muscle fibers.
Okay.
There. Oh yeah. About Hansen's disease, leprosy. It's a horrible disease, obviously, and it's interesting that people have known for a long time that it's contagious, and so there are all these prescriptions and, proscriptions and ways of keeping people that have leprosy away from everyone else. So we have them in very ancient writings, including the Bible, where it says lepers are supposed to wear a cloth over their upper lip, which suggests they even knew that it was like the droplets of water in saliva that could transmit it. And it was really bad to have leprosy because nobody would have anything to do with you again. You had to go around begging for food, etc. The irony is that 95% of the population can never possibly get leprosy, right? Almost everyone is immune. It's still spread through aerosol droplets, but only 5% of the population will ever respond to it. And so all those thousands, hundreds of thousands of people who suffered from Hansen's disease in the past, who were made, lepers, they had to stay away from everyone else. That was almost certainly unnecessary. And today, as I said, it can be treated with antibiotics. Okay, now back to the confusing thing that some sympathetic cells are cholinergic. Done.
There's a chance that the fact that I get exiled out with leprosy from society led to 95% but people who quite with it. Maybe it was 50% back then. We'll get rid of all of them.
So you're saying maybe they were, applying some selective pressure to favor those, but it seems to me that the way to make sure fewer and fewer people who are susceptible are in future gene pools is to make sure everyone's exposed to it. So I don't know. And all this stuff about it, let's see, the bacterium wasn't isolated until the early 20th century. And well past that, they were still, kept isolated. And the fact that most people are immune to it, that's very recent news. People haven't known that very long. Okay. Some sympathetic cells release acetylcholine, and the big exception are sweat glands. So it turns out that the neural crest cells, in the beginning, they're all cholinergic. And then some settle in the sympathetic ganglia, and they then stop making acetylcholine and start making norepinephrine. And then they send their axons out to innervate the body, to mostly give that fight or flight response. But a subset will end up innervating sweat glands, and then they go back to being cholinergic, because it turns out it's cholinergic drugs that activate sweat glands and cholinergic transmission. So that's what it says there. So the question is, what's driving them to switch back to being cholinergic? And the answer is the targets. The sweat glands are going to instruct the sympathetic fibers that reach them, okay, stop making norepinephrine. I need you to make acetylcholine. And the reason we know the targets are sending this information is because of experiments that were done where, I don't know if you know this or not, but rats do not have any sweat glands on most of their body. The only place rats have sweat glands are on their paws. And so when rats need to lose heat, like dogs, they need to pant. Or if they're really distressed, they may lick their paws and have the evaporative loss there help cool things off. All right. And so in these experiments, they could take this glabrous skin and transplant it on the body of a different pup. And when it grew up, the question was, are the sweat glands there going to be receiving norepinephrine, noradrenergic stimulation, or cholinergic? And the answer is cholinergic. So the cells switched their fate again. They were cholinergic, then they became noradrenergic, and then once they reached the sweat gland, they got some message that says, go back to being cholinergic. And that's how it works. questions about that. They also did the reverse, where they took a bit of back and put it onto a paw. And so I haven't seen pictures of it, but I guess they had these rats with hairy paws. Had nothing to do with masturbation. That's a myth. Okay. Oh, sorry, wrong class. All right. Another quiz. That's what we need. So, as you can see, we're not going to finish chapter 4 today, but we are going to finish it by next time. And so we will start chapter 5 on Thursday. And the notes for that are up already. So take a look. And I want to talk some more about neuronal fate in the cerebral cortex. And this takes advantage of the fact that we established earlier that there is a regular pattern of birth dates, in that the earliest born neurons, I'm going to ignore for a moment the tiny number that end up in layer 1, the molecular layer. But after that, layer 6 gets formed, and then cells migrate past layer 6 to make layer 5, then 4, then 3, then 2. So that if you know the birth date, if you know the right species, and you know the birth date of a cell, you have a pretty good idea of which layer cerebral cortex it's going to end up at, right? So the earliest born are going to end up in layer 6, and the later born are going to end up in layer 2. And as they kick out of mitosis, they end up with different factors, different places along that line. And someone asked, how do the cells know which fate they're, and they will take a different fate depending on whether they're in layer 6 or layer 2, right? The neurons in those two different places have very different fates. And so someone asked, what directs the cells to be in the layer they're going to be in and take on the fate they're going to be in? Is it that they know, based on what stage of development they kick out of mitosis, where they're going to be? Or do they leave the subventricular zone with no idea of what they're going to do until they shinny out along the radial glia, and they just follow a simple rule? If there's some neurons here already, keep moving and keep settling further and further out so that you have that inside-out pattern. And the answer is yes. Both of those things are happening. And it was some pretty clever experiments that showed that. And I think we just have time to talk about them. So these experiments were done in ferrets. And ferrets are born with their eyes closed, so they have a relatively immature nervous system at birth, like rats, right, and cats, but not like guinea pigs, for example. And the researchers took advantage of this, where they learned, first of all, that the inside-out pattern is there, that if they give thymidine on the day of birth, there's still some neurons that are dividing, some cells that are dividing and forming neurons, and so they'll be labeled with the thymidine, but they all end up in layers 2 and 3. Whereas if they gave the injection earlier, on the 29th day of embryogenesis, then those cells all ended up in layer 6. Everyone get that? And then they did a clever thing where they injected animals early in development, and then they sacrificed the animal and harvested some subventricular zone from these young ferrets and planted them into a newborn ferret. So they refer to these as heterochronic transplants. Do I have that? Because there are cells of different ages there, right? Some are from a young ferret and some are from a newborn ferret. Got that? And they can tell which is which because they labeled the young neurons with the thymidine. And so the question is, where will they go? If we'd left them in the ferret fetus, they would have ended up in layer 6. But we put them into an older ferret, where the cells now are making layers 2 and 3. Where will they go? And the fascinating result was either or. Some of them indeed went to layer 6 and took on the fate that they would have had if they had been left in the younger ferret. But others of them went past layer 6 and changed their fate. They became neurons in layer 2. And happily, there were very few neurons that ever got off anywhere in between. So it's like there are two minds. One, I'll make layer 6 because somehow I know I'm in a young ferret, and that's what I ought to do. Whereas other of the cells, I have no idea what age the ferret is that made me, but I'm looking around and, hey, layer 6 is occupied, layer 5 is occupied. I'm going to go past those and go out to layer 2. See that? And it turned out that there was a simple thing going on, that if they were transplanted after their last S phase, the last time they're taking in thymidine in preparation for that final division, when they were transplanted in that state, then they would get off at layer 6, as if somehow in that last division in the subventricular zone, they got some sort of instruction: we're a young ferret. You go be layer 6. But if they got transplanted after the last S phase, it was like that made them plastic. We're ready to go elsewhere. How's that? And then I do want to get this. So there is this mixture of remaining plastic and not. And again, it seems like that final mitosis is an important watershed event that's going to tell them whether they should remain plastic or, okay, it's time for you to grow up now. You know, it's like when you become 18 and your parents say, no, you got to leave now. So you've got to make a decision, college or the work gang or whatever, right? And there. The other thing I want to show you is this, just because I want you to understand this is kind of a joke. The caption could read something like, here's a simplified diagram of transcription factor interactions in a regular cell. And it happens to come from people who were studying stem cells, but each one of these represents a different transcription factor, okay, who influence the expression of other transcription factors. And these are the pathways that have been mapped out so far, right? So no, I don't want you to remember this. I just want you to know that it's incredibly complex, which tells you a couple of things. Number one, natural selection does not care whether you understand things or not, right? These things happened through the random process of natural selection, and not for the convenience of people to try to understand them. And therefore, it's a lot of work to try to understand them. And to the extent that we know anything about these sort of interactions, the secret is to find a simplified system and to find a way to isolate one particular signal so that you can study it and manipulate it and get some idea about what happens. Okay. Next time, we'll talk about why your brain wasn't bigger than it was when you were born, and why you undergo fetal development. Why, until you were about 3 or 4 years old, you were just a fetus outside mom's womb. Okay, talk to you then.
You really need to search Bride.
Hi, Kennedy.
Please. Good team. You have minutes.
I do.